Hello and welcome to the cell cycle review. So in this review we are going to take a look at a few key processes that are very important for your understanding. The first is really just the purpose of mitosis. What is the purpose of this division process? And in saying that, that's really one of the major purposes, to make more cells for the cell to divide. And then also some key terminology involved in the process. And then talking about the specific stages and events of the cell cycle. And then looking at different mechanisms for control of the cell cycle. And then we'll have a short discussion on the development of cancer as a result of problems with the cell cycle. All right, so basic purpose of cell division in an organism that is unicellular or made of one cell most often will be just to reproduce. So bacterial cells, some eukaryotic organisms that are unicellular reproduce in this way. But for multicellular organisms like ourselves, we use this cell process this process of cell division to replicate cells and to grow and repair cells that are already in existence. Some key terms. All of a cell's DNA is called its genome. So in our cells we have 46 chromosomes, 23 pairs. We'll get to that idea of pairs when we talk about meiosis, but all of that is the genome. And the genome, of course, like I said, made of chromosomes. In humans it would be 46. And each of those chromosomes is made of the DNA molecule, the double helix, and that DNA is wound around certain proteins called histone proteins that allow it to be packaged into this complex of DNA and proteins called chromatin. And on the DNA molecule, certain segments of the DNA are called genes, and those genes code for particular proteins. That's the basic idea. All right, and then we have our cell division process. We have our chromosome shown here in this singular unit. Of course, that would be the DNA double helix and the histone proteins that the DNA is wound around. And during the division process, each chromosome makes an exact copy of itself, and it's connected at this area called the centromere. During the division process, the chromosomes split, and once they split, there are chromosomes in their own right, and they go to the two new cells, or daughter cells. Now, when they're connected, we call these sister chromatids. That's very important. They're sister chromatids, and it's one chromosome when they're connected. All right, cell cycle, there's two major phases. We have interphase and the mitotic phase. So interphase, as you can see, this is a really great representative picture of the time that the cell spends in the different phases. Now in interphase, the cell is preparing for the division process. So a lot has to occur here. The cell grows, the DNA is synthesized, we make all of those sister chromatids, and we are preparing, getting all the molecules ready for this wonderful mitotic process. So there's interphase with all of its subphases, and then prophase, metaphase, anaphase, telophase, and then finally at the very end the cell divides. The cytoplasm is cut, cyto-cutting, kinesis, to cut. And as far as the different stages are concerned, you're going to want to know the major events in each stage. And in this series of slides, it really does show you all of these, or spell out for you all of these different events. So take a look at the PowerPoint. These are exactly from the PowerPoint for this chapter. And then also be able to recognize the cells, the actual human cells I have along with those pictures. So this would be an example of a cell in interphase. You see that kind of cloudy, you can't really see the actual chromosomes in their condensed form. And the same goes for all of these other stages. So you want to know the particular events and then of course the order in which they occur. And again, each of these has particular events, and then of course the example that I have with each. Make sure you can recognize each of those. All right. Okay. Well, one thing here I want to look at is the spindle and how chromosomes, sister chromatids, after they separate, how the chromosomes move to opposite poles during anaphase. And an important thing to understand is that they actually walk down this which was the sister, now is a chromosome, walks down the spindle fiber to the opposite pole. Okay? And as it walks down the spindle fiber disintegrates behind it. There's a really nice experiment, and there are a series of pictures that look just like this, in which they discovered, that the chromosomes actually walk down the microtubule or the spindle fiber as opposed to being pulled from the poles. So take a look at that experiment. And then the process of division in, like we had said, the prokaryotes is outlined here. It is called binary fission. That one chromosome, it's a very big circle, it's kind of bunched up here in the center, is copied, and the division process is pretty straightforward. So take a look at that. And then moving on to how the cell cycle is regulated. So there are molecular control signals inside the cell. The one we're going to focus on here, there are actually several checkpoints in the cell cycle, but we're going to focus on something called the G2 checkpoint. It's the checkpoint that comes right at the end of interphase, and you need a molecule in order to, at the end of this G2, in order to get into the mitotic phases, in order to start the cell division process. And this diagram outlines those molecules, or the molecule and the parts of the molecule that are involved. So we have something called a cyclin-dependent kinase, and we have cyclin. Okay, so cyclin, the cyclin part cycles. It accumulates towards the end of interphase. It's used during mitosis, and then it breaks down at the end of mitosis. Whereas the cyclin-dependent kinase, it's always present in the same amount. It's constant. And the cyclin-dependent kinase, it is dependent on cyclin for its activity. And the two together are called maturation promoting factor. I like to call it mitosis promoting factor because you need it in order to get into mitosis. If you don't have this molecule, no mitosis for you, because it catalyzes, or it allows the reactions of mitosis to occur. Formation of the spindle, the chromosomes moving to opposite poles, dismantling of the nuclear membrane. All of those events of mitosis require this molecule. Okay, this is just another diagram. This is actually in your assignment for this week, and I ask you to draw a line where cyclin-dependent kinase would most likely be. It shows the levels of cyclin. So remember, cyclin cycles, so it increases towards the end of interphase, peaks in mitosis, and then falls. Okay, increases, peaks, and then falls. So you would expect the cyclin-dependent kinase, being as though it is always present, to have a certain conformation in this graph. So I will, or a certain view in this graph. So I want you to think about that and complete the assignment question based on that idea. Now, on to cancer. So cancer cells have a problem with the cell cycle. There's something wrong with the checkpoints, for instance. So perhaps the cell doesn't have enough of MPF, but for some reason there's a problem with the cell and it just doesn't listen to that control mechanism, and it goes on through to mitosis and it can't complete the events correctly. So we have problems that occur, and the problems that can occur is the cell divides when it shouldn't, and we get out of control cell growth. So out of control cell growth can result in tumor formation. Benign is really the least risky, or least, I could say, deadly, if you find it early, of course. And a benign tumor can develop into a malignant tumor if left unchecked or it is not found. And then, of course, a malignant tumor can develop into something called a metastatic tumor. And we're going to take a look at this process, this development or this progression, I can say more likely, of tumor or of cancer with looking at breast cancer. So we have our benign tumor. Benign tumor is a ball of cells that's nice and tightly adhered together, and it's really just in one tissue or one organ. As you can see here, now if left undetected, it can develop into a malignant tumor where it's less, the cells are less adherent to one another and they're more diffuse through the tissue or the organ. It'll be much more difficult to remove this type of tumor growth with a surgery. When the surgeon goes in to remove the tumor, these cells are not red in color, so the cells look very similar to the healthy cells. So what will usually happen is the organ is removed at this point, and oftentimes, even in a benign tumor situation, the organ is oftentimes removed in current times. Now, the malignant tumor can develop further if left undetected and start to break off pieces of it, cells in particular, and move through the blood vessels or the lymph system and travel to other organs. So this is called metastasis, or the tumor metastasizes. So you can have breast cancer with metastasis to the lungs or to the brain. Those are really common sites of metastasis. So you have one single cell land in the lung, and it is out of control. The cell does not listen to the cell cycle control mechanisms, and tumor development results. Okay. So benign being the having the best prognosis, malignant kind of in between, depending on the type of malignancy, where it's located, what organ it's located in, and then of course metastatic being having the poorest prognosis. Okay, so that is our review of Chapter 12. I hope that this has helped you better understand this cell cycle, and you please, by all means, ask if you have any questions about these topics.